Hello friend, all are welcome to my channel Get Easy Medical Micro. Today we are going to discuss about the bacterial spore. So what is bacterial spore? A number of gram-positive bacteria, those of the genera included in Clostridia, Bacillus, they are forming a resistant dormant structure in environment. That condition is called the spore, and it is also called the endospore also. So again I am repeat, a number of gram-positive bacteria, those of the genera Clostridium and Bacillus, they are forming a resistant dormant structure. This process is called the bacterial spore or endospore. So what is the reason of the endospore formation? They develop endospore in unfavorable condition. They are try to exist their body. That time they will form endospore. So what is the reason? One is the lack of nutrition. When the bacteria unable to take nutrition from the environment, that time they will produce endospore. Similarly, when they get any unfavorable condition, okay, that time also they are forming endospore or bacterial spore. Okay, the two reasons: unfavorable condition, another one is lack of nutrition. Okay, so nutrition depletion, also called nutrition, they are forming spore or develop for lack of nutrition or depletion of the nutrition. Okay, so one causes is causes depletion of nutrition. Another one is any unfavorable condition. When they are unable to survive, that time they are forming endospore, try to exist their body. Okay, that time what happened? That time no other external environment destroy themselves. Okay, so it can generate another bacterial cell also when they will get proper environment or proper nutrition. Okay, so one single bacteria, living bacteria, can form resistant structure, endospore, and when they get favorable condition, they also transform a one single vegetative cell or a single living cell. Okay, so bacterial spore, single spore, living bacterial cell, they can transfer a single spore. Okay, similarly, this process is called the sporulation. This process, the formation of spore, okay, it has one process we'll discuss. This process is called the sporulation. Similarly, one single spore can form a one single vegetative cell or living bacteria. This is process is called the germination. Okay. This is the sporulation. This process is called the germination. Clear? Now comes to the how bacterial spore will form. This is the process. We will discuss it. The process. At first, when they get unfavorable condition or lack of nutrition or depletion of the nutrition in environment, they are unable to take nutrition, proper nutrition from the environment or any external matter hampering to live their life. That time they are forming spore. So how they will form? At first, a single bacteria looking like this single bacteria. Okay, this is the normal cell. Normal cell. Okay, normal cell, it will replicate at first and form a replicate at first. This chromosome, not the whole cell, this chromosomal part or DNA at first replicate at first and form a daughter DNA. This is the replication. Okay, first this is the daughter DNA. Yeah, this is the replication. At first it will form two separate DNA. Okay, this is the replication process. Next, after replication of the chromosome, it make a septa like this. This is septa, means separation. This is called spore septum. This is called spore septum. Okay, this one is daughter DNA, this one is parent DNA. Okay, after that, after the separation of that both chromosome, the one means daughter chromosome goes towards the edge of the cell, towards the edge of the cell, because before formation of the spore, it take nutrition from their own cell and form an external layer. That's why they have to collect so other chemical molecule and etc. So that's why they goes towards the edge of the cell. Okay, this one is the layer septa. Okay, this process is called the forespore. 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 Next is this chromosome part. Okay, covered with a single layer like this. This portion is called, this one is septa, spore coat. Spore coat, make a coat or layer. around the daughter DNA. After that, it form another layer. It form another layer. Okay, form another layer. This one is single layer. Okay, and this one the second layer. Okay, this one is the second layer. Okay, so after formation of the second layer, it will be more resistant in nature. So no other external matter can destroy the spore. Okay, so this process is called the spore cortex. Okay, so after formation of spore cortex, it normally release a lysozyme enzyme, release a lysozyme enzyme and brush the cell and release the spore. So this one is the, suppose like this. Okay, this one is the spore. Okay, so this one is released from the bacterial cell. Okay, so this process is called the exosporium, the release of the spore from the cell. That time it will release the lysozyme, okay, and brush the cell wall and release the spore. This is called the spore, original spore, and it destroy the main cell or parent body. Okay, that is the process. Okay, at first normal cell it can replicate and form a two separate chromosome. This is the daughter DNA and parent DNA. After that it form a spore septum, form a septa and different between the daughter and parent chromosome. Then it goes towards the daughter DNA, goes towards the edge of the cell to collect extra chemical compound from cell wall. This is called the spore. Then it form a coat or layer. This called is spore coat, and it form another several layer or two layer. It is called spore cortex, and it form a more resistant structure. Okay, after that it release a lysozyme and brush the cell, and spore is come out from the cell and the main cell will be destroyed. This process is called the exosporium, particular process, and this total process is called the sporulation. That is the formation of spore. Formation of spore. Okay, this is the sporulation process. Similarly, I also say the when we will treat lysozyme or it get favorable condition, that time the cortex part will be absorbed and form a normal general cell. Okay, or normal vegetative cell. This process is called the germination. Okay, So now we are discussing another things. If we cross section the spore, we will see this one is the okay, this one is the spore core. Okay, this one is the spore cortex, inner membrane, and this one is the spore coat. Okay, and exosporium. So it is the another formation of exosporium. After the formation of this, it secrete the lysozyme and brush the normal cell. Okay, so this portion is together. This is the cross section. Cross section of spore. Okay, so this structure, if we cross section, then we can observe this all layer. So this one is very important. This pore core and this pore coat. Okay, when bacteria will germinate, that time it will easily absorb in the environment, or when we treated the lysozyme externally, that time it easily destroy. Okay, and we can transform a normal bacterial cell. Okay, or this is called the germination processes. Okay, so this much is helpful for you. Thank you.